Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. May all the peace and blessings of the Almighty Allah subhanahu wa ta'ala be on all of us. Let's begin our class. Now today, as you can see, I want us to dive and navigate through the osmosis. And osmosis is one among the three physiological processes being discussed in cell physiology, a chapter in biology form 1. Now class, I've not written the definition of osmosis, but we can just say before we start the terms and the pressures associated, as well the roles. Now class, what is osmosis? And what are you supposed to be knowing when defining osmosis? Osmosis is a physiological process. And osmosis being a physiological process class, it basically involves movement of water molecules from their region of high concentration to the region of low concentration, or rather from the region of high water concentration to the region of low water concentration. And therefore, we can define osmosis as the process by which water molecules move from a region of high water concentration to a region of low water concentration through a semi-permeable membrane. Now, today I want us to look at the pressures associated with osmosis, terms used in water relation in animals as well in water relation in plants. We also want us to look at the role of osmosis. Now, but before that class, I want us to understand that the types of solutions we have and where does water molecules move from and from and to. As you can know class, there are three different solutions, and that is we have the hypotonic solution, lowly concentrated solution, or rather a solution which has high solvent molecules and less solid molecules. You have a hypertonic solution, sometimes referred to as a concentrated solution, and have high solute concentration and less solvent concentration. And we have an isotonic solution, a solution in which both the solute and the solvent are at equal measures, hence equilibrium. Now class, let's look at the pressures associated. And we know that water moves from hypotonic solution to hypertonic solution, or rather from the lowly concentrated solution to the highly concentrated solution through the semi-permeable membrane. Now before that, let's look at the pressures associated. Now the first pressure class is called the osmotic pressure. Now, what is an osmotic pressure? It's a pressure applied against a semi-permeable membrane. These are semi-permeable membrane, right? Now, osmotic pressure class is a pressure applied against a semi-permeable membrane. Now, what does this pressure do? It stops water molecules from the distilled water into entering a concentrated solution. In a nutshell, osmotic pressure basically means a pressure that is against osmosis from taking place, or rather stops osmosis from taking place. Very good. It's a pressure that stops osmosis from taking place, or rather, you can define as a pressure applied against a semi-permeable membrane to stop water molecules from distilled water into entering a concentrated solution. Now let's look at another pressure known as osmotic potential. Now osmotic potential class is the hidden pressure that only becomes real when a concentrated solution is separated from distilled water by a semi-permeable membrane. It only becomes real when the concentrated solution and the distilled water are being separated by a semi-permeable membrane. And as such you say, it is the hidden pressure of a concentrated solution that only becomes real when the solution is separated from the distilled water by a semi-permeable membrane. Now, it is the hidden pressure of a concentrated solution that only becomes real when the solution is separated from the distilled water by a semi-permeable membrane. Let's go to the next pressure. It's a target pressure. A target pressure is an outward pressure exerted by the vacuole as a result of the water is drawing in when placed in a hypotonic solution. And therefore, class, we define Tagger pressure as an outward pressure exerted by the enlarging vacuole as it gains more water when placed in a hypotonic solution. Let's go to the fourth pressure and last pressure, wall pressure. Now, class, we know that Tagger pressure is a pressure exerted outwards, and therefore there is an opposite and equal pressure which acts inwards to stop the effect of the Tagger pressure. And therefore, wall pressure is an inward pressure that is opposite and equal to Tagger pressure. And therefore, and that is briefly about the pressures. Now let's look at this setup. Here we have distilled water made up of water molecules. You can see we have the semi-permeable membrane, and these are the pores of the semi-permeable membrane. We also have sugar crystals making up sugar solution. Now, this one is the concentrated solution, right? And this is the dilute solution. And therefore, we said the osmosis is the process by which water molecules move from the region of high water concentration to the region of low water concentration, and as such, therefore, water molecules will move from this region and will enter into the sugar solution based on the definitions given. Now, let's go to the terms used in osmosis. Now we have hemolysis, threnation are the terms used in water when discussing water relation in animals. And therefore, hemolysis comes when when animal cell is placed in a hypotonic solution, while threnation comes when an animal cell, an example given is the red blood cell, is placed in a hypertonic solution. Now, in each case, what happens and how can we define the processes? Now, we say hemolysis is the process of bursting of animal, or rather, a given example is the red blood cells when placed in a hypotonic solution. As a result of the water it draws in, it bulges outwards and eventually bursts. Now, threnation is the process of shrinking of animal cells, e.g., the red blood cells, when placed in a hypertonic solution as a result of water loss. You know what happens when they are placed in those two different solutions. Now, turgid. When can we say that a plant cell is turgid? A plant cell is said to be turgid when the Tagger pressure equals to the wall pressure, and therefore the plant cell becomes firm and rigid, and it also attains the turgidity, and hence the plant is said to be turgid. It's also associated with when a plant cell is placed in a hypotonic solution. Let's come to the flaccid. Now, flaccid class is the state where the cell becomes soft and less rigid when placed in a hypertonic solution. Now, the process is called plasmolysis, right? But now the state of the cell is what is described to us as flaccid. Now, the state where the cell becomes soft and less rigid or flabby is what we call flaccid. Now, let's go to the plasmolysis. It's the process by which cell membrane shrinks and pulls away from the cell when plant cell is placed in a hypertonic solution. Very good. When a plant cell is placed in a hypertonic solution class, the cell membrane pulls away from the cell and shrinks, and therefore, as a result of water loss to the hypertonic solution surrounding it. Now, deep plasmolysis is the opposite of plasmolysis, where we reverse plasmolysis by placing the flaccid cell in distilled water. And therefore, we say it is the reverse of plasmolysis, where the flaccid cell is placed in distilled water. Now, let's look at the role of osmosis in animals. Now, role number one, we have absorption of water from the soil. Now, this role is principally played by the roots of the plant. And therefore, water which is in high concentration in the soil is being absorbed from the soil through the process of osmosis into the root hair cells. Now, this water moves successfully through into the xylem of the root. From the xylem of the root is conducted to the xylem of the stem, where it is conducted to the xylem of the leaf, and also it gets out of the leaf through the process of transpiration, and therefore the cycle begins again, and that is discussed in great details when we are discussing about absorption of water from the soil and a transport in plants in biology form 2. Let's go to support. How can osmosis bring about support? We say when plant cells absorb water, they become firm and rigid. And when a plant cells become firm and rigid class, meaning the plant has already gained support. Very good. Let's go to opening and closing of the stomata. How does the process of osmosis bring about the closure and the opening of the stomata? During the day class, what happens is, during the day, in the presence of the sunlight, the chloroplast in the guard cell absorb sunlight energy and they photosynthesize. And once they photosynthesize class, they manufacture sugar. And sugar is osmotically active, and it will absorb water molecules from the neighboring adjacent epidermal cells. And once it absorbs water from the neighboring adjacent epidermal cells, it is going to bulge outwards, hence the opening of the stomata. And at night, however, when there is no sunlight, the process of photosynthesis won't take place, and therefore the sugar that accumulated during the day is exhausted, and therefore that means the osmotic pressure of the guard cells will fall down, and as such, therefore, water will be lost from the guard cells into the neighboring adjacent epidermal cells. The guard cells shrink, and hence the stomata closes. Now let's look at the feeding in insectivorous plant. These are plants that suddenly trap the insects because they use the nitrates in them, and therefore what happens is when an insect lands on an insectivorous plant, they suddenly change their target, and once they suddenly change their target, the leaf closes, and once the leaf closes, they digest the insect and they use up the nitrate. Let's go to the osmoregulation. Now, how does osmosis bring about the osmoregulation? Class, we know that in the kidney tubules of animals, there is water reabsorption along the loop of Henle, the descending loop of Henle. And therefore, class, when the water is reabsorbed from the kidney tubules across the tubular walls into the bloodstream, that water absorbed into the bloodstream is used to regulate the osmotic pressure of the blood, and hence, therefore, osmosis. And you know that the water has been absorbed through the process of osmosis from the tubular walls into the bloodstream, and it has regulated the osmotic pressure. And therefore, we say that the process of osmosis plays a significant role in osmoregulation. And therefore, class, osmosis is a very basic concept, and it is used, and osmosis basically takes in our body cells where water molecules form from one cell of the body to another cell through the process of osmosis. And in our next lessons, we will be looking at other issues that require deep analysis. For instance, where we have a scenario where we give you a setup, where we have a certain solution in a whisking tubing and another solution in the beaker. Now you will be told what type of membrane does the whisking tubing represent. The whisking tubing represents the semi-permeable membrane. Another question, which type of cell does the whisking tubing represent? It basically represents the cell membrane. And therefore, you just analyze where will water move from and into. And therefore, you can give observation as to where, as to why, as to whether the whisking tubing will expand or it will shrink, depending on the movement of water from where to where. And therefore, wish you all the best. Have a blessed and a prosperous day. With you here is Teacher Muhyiddin Khalif. Will demystify the concepts that give you nightmares. Stay tuned and pray for the teacher to get paradise. Wish you all the best.